The research article that I will be presenting on today is Polymerized Human Hemoglobin Increases the Effectiveness of Cisplatin-Based Chemotherapy in Non-Small Cell Lung Cancer. My name is Sophia Brooks, and this presentation was prepared for Biology 4366-600 or Cancer Biology at Texas A&M University, Kingsville. So, non-small cell lung cancer, which is commonly abbreviated as NSCLC, is one of the leading causes of cancer death. It is defined by cancer.gov as a group of lung cancers that are named for the kinds of cells found in the cancer and how the cells look under a microscope. It is the most common type of lung cancer that can affect different parts of the lungs based on the subtype. There are three main subtypes of non-small cell lung cancers, which are adenocarcinoma, squamous cell carcinoma, and large cell or undifferentiated carcinoma. So adenocarcinoma usually starts in cells that are primarily used for secretion. This usually will spread from the outer part of the lung into the center, so it's more likely to be found prior to spreading. Squamous cell carcinoma is known for starting in squamous cells found on the interior of the lung airways. They're usually found near bronchus in the central part of the lungs. Large cell or undifferentiated carcinoma is not known to initiate at any part of the lung. It can start anywhere and spread quickly, which can make treating it very difficult. This is usually the most difficult subtype to treat. And while there are other subtypes, these are the most common. So non-small cell lung cancer makes up 80 to 85% of all lung cancers, and by the time it's diagnosed, about 40% of the patients have already progressed to stage 4, which can make it difficult to treat. It is the leading cause of cancer death. Localized non-small cell lung cancer means that the cancer has not spread outside of the lungs. The 5-year survival rate for individuals who are affected by this is 61%. People with regional non-small cell lung cancer or cancer that has spread outside of the lungs and into nearby areas is about 35%. For the people with metastatic lung cancer or cancer that is spread outside of the lungs and into distant parts of the body, their survival rate is about 6%. While these rates can seem extremely disheartening, it's important to note that these rates can vary depending on the individual, the stage, and the type of lung cancer. Some patients can live for many years after being diagnosed, and the cancer can be cured in certain situations. Screening for lung cancer is completed using CT or CAT scans. These scans photograph the inside of the body using an X-ray machine. These detailed photos are later combined into detailed three-dimensional images that show tumors and abnormalities. So the symptoms of non-small cell lung cancer are fatigue, cough, shortness of breath, chest pain, loss of appetite, coughing up mucus or blood, unintentional weight loss, and hoarseness. Non-small cell lung cancer is diagnosed using the CT, PET scans, and MRI scans. And treatments for non-small cell lung cancer will vary based on the individual and the doctors that treat them. The most common treatments that are used is surgery, radiation, chemotherapy, targeted therapy, and immunotherapy. The treatment type that we'll be focusing on today is chemotherapy using cisplatin. So this research comes with a great deal of terminology that most people who are not experts in this field are not familiar with, starting with NSCLC, which, as we previously discussed, stands for non-small cell lung cancer. Cisplatin, as we also covered, is a chemotherapy medication that is used to treat various types of cancers. It's the main treatment that is used in this study, in combination with poly-HB, or polymerized hemoglobin. Hypoxia is a condition where the environment is deprived of oxygen supply. Hypoxia-inducible factor 1 alpha is a regulator that sustains the survival and growth of cancer cells in a hypoxic environment. ROS stands for reactive oxygen species, which are unstable molecules that can contain oxygen. They interact with the other molecules that are inside of the cell, and they can lead to cell death if they build up inside of the cells. And the IC50 refers to the point where only half of the initial value of cells are viable. We'll see this in the resulting graphs. And then the Western blot is a lab method that can be used to detect specific protein molecules in a sample. It can highlight certain proteins so that they can be differentiated from the other proteins around it, and they can show like the size of the protein, or like in this experiment, it shows the amount of the protein expression. So you'll again see this in the results. So the pertinent information for this study starts with hypoxia. As we talked about earlier, hypoxia is a condition where the environment is deprived of oxygen supply. While hypoxia can affect both large and small environments, the study focuses on hypoxia in a tumor microenvironment. Cisplatin chemoresistance is commonly associated with hypoxia in the tumor microenvironments. The goal of this study is to reduce hypoxia in the microenvironment and therefore reduce cisplatin chemoresistance and reduce the amount of non-small cell lung cancer present. So, polymerized human hemoglobin is an oxygen carrier. It was originally developed to treat anemia, but it has also found use in treating hypoxic tumor microenvironments. So, while cisplatin is known for the formation of mitochondrial ROS that lead to the initiation of apoptotic pathways, hypoxia-inducible factor 1 alpha is known to reduce the amount of ROS in cancer cells, promoting cancer cell survival. When hypoxia-inducible factor 1 alpha is active, or there is moderate hypoxia, there are a bunch of intracellular pathways that are activated which cause the cells to survive. When hypoxia-inducible factor 1 alpha is active, also the cells begin glycolytic synthesis of ATP and increased synthesis of vascular endothelial growth factors, which causes angiogenesis in the tumor's microenvironment, and it allows a lot more blood to reach the tumor and it promotes cell growth. So the expression of hypoxia-inducible factor 1 alpha is very likely to affect the effectiveness of cisplatin in that hypoxic tumor microenvironment. So cisplatin treats the cancer by cross-linking and damaging the DNA, which results in apoptosis. It promotes the formation of mitochondrial ROS, which can also promote the initiation of the apoptotic pathways. It does this by damaging the membrane of the mitochondria, among other cell damage, but that's the main one. By increasing the amount of oxygen in the tumor microenvironments, cisplatin can induce the apoptotic pathways and kill the cancer cells. The whole point of this research experiment is to see if polymerized hemoglobin is an effective way to increase the amount of oxygen in the tumor microenvironments and therefore decrease the cisplatin chemoresistance in non-small cell lung cancer cells. So by polymerizing the human hemoglobin in the relaxed quaternary state, the oxygen affinity was significantly increased. The effective hydrodynamic diameter was also significantly increased, and there was definitely slower rates of autooxidation in comparison to the unmodified hemoglobin. So in this figure, we can see the ROS and the DAPI stains for polymerized hemoglobin and a vehicle control. So that's in figure A, and figure B will show the ROS fluorescent intensity of the digested A49 cells following exposure for 24 hours to the polymerized hemoglobin and the vehicle control. So after the 24-hour incubation, the A549 cells with the polymerized hemoglobin, there was a significant increase of expression of ROS in comparison to the control group, which we can see here. So this figure shows the effects of the polymerized hemoglobin and the vehicle control on the expression of the hemoglobin-inducible factor, oncogene P53, and actin using the Western blot, which we discussed earlier. So following the incubation with the polymerized hemoglobin, we can see that there was a reduced hypoxia-inducible factor 1 alpha expression and P53 oncogene expression. So this figure shows the tumor cell viability in culture measured by the percentage of the live cells after treatment with 3 treatment groups for the 3 different types of cells, which are H24, H416, A549. So for the H23 cells, there was not a significant increase in the number of necrotic cells when it was treated with any concentration of cisplatin with polymerized hemoglobin when compared to the cells treated only with cisplatin. However, there was an increase that was shown in the cells undergoing apoptosis and the cells that were treated with polymerized hemoglobin and cisplatin at a concentration of 0.2 grams per deciliter when this is compared to the polymerized hemoglobin and cisplatin at a concentration of 0.1 grams per deciliter in the control group. There was an increase as well in the number of the co-stain cells, or the cells that were necrotic and undergoing apoptosis, when there was a presence of polymerized hemoglobin with the highest amount and cisplatin and the polymerized hemoglobin with a concentration of 0.2 grams per deciliter. So for the H460 cells, there was a significant increase in the number of apoptotic and necrotic cells when there was a presence of polymerized hemoglobin. So while the concentration of polymerized hemoglobin does not appear to affect the number of apoptotic and necrotic cells, when they are evaluated separately, there's a significant increase in the co-stain cells when polymerized hemoglobin is present. So for the cells that are in the presence of 0.1 gram per deciliter hemoglobin, there's an increase. And for the A549 cells, there was very similar behavior to the H23 cells. The polymerized hemoglobin did not change the number of cells that were positively stained for necrosis in comparison to cisplatin-only treatment. The presence of the polymerized hemoglobin did increase the number of cells that were undergoing apoptosis when you compare them to the cisplatin-only treatment. And the increase did not seem to correlate with or depend on the concentration of the polymerized hemoglobin. The highest number of viable cells were found in the cisplatin-only treatment group, just the same as the other cells. The cisplatin in combination with 0.1 grams per deciliter polymerized hemoglobin rendered the smallest number of viable cells, but all of the differences for the viable cells were significantly different. And so, moving on. So this is figure 3A. In this figure, it shows that for the H23 cells, there was a significant decrease in the cell viability following exposure to the cisplatin and the polymerized hemoglobin at different concentrations. So only half of the cells were viable after treatment with cisplatin at 2.85 micrometers, half were viable after treatment with cisplatin and polymerized hemoglobin at a concentration of 0.1 grams per deciliter, and half were viable after treatment with cisplatin and polymerized hemoglobin at 0.2 grams per deciliter and a concentration at 0.8 micrometers. And so in this figure, we can see that for the H460 cells, half of the cells were viable for cisplatin at 1.31 micrometers, and then half were viable for cisplatin and polymerized hemoglobin at 0.1 grams per deciliter, with a concentration at 0.66 microliters. So this is what I was referring to when I said the IC50. In the study, it refers to the half point as the IC50, but here I'm just saying the entire thing. So half of the cells, or the IC50 for the cells, for cisplatin and polymerized hemoglobin at 0.2 grams per deciliter was at a concentration of 0.37 micrometers. So this figure shows that for the H549 cells, half of the cells with cisplatin were viable at 5.27 micrometers, half of the cells were viable for cisplatin and polymerized hemoglobin at a 0.1 grams per deciliter concentration at 2.47 micrometers, and half of the cells were viable for cisplatin and 0.2 grams per deciliter polymerized hemoglobin at 1.31 micrometers. So from the results that were shown in all three sections of this graph, we can conclude that the largest decrease in the cell viability during hypoxia occurred after treatment with cisplatin and polymerized hemoglobin at 0.2 grams per deciliter concentration, which was the highest amount of polymerized hemoglobin. So the graphs in this figure display the changes in tumor size for the mice that were implanted with H23, H460, and A549 cells. This was followed by a treatment with cisplatin in combination with polymerized hemoglobin, and of course the vehicle control, which was 0.9 saline, if I'm not mistaken. You can see that regardless of the treatment that was given, the different types of tumors had different growth rates. So H460 displayed the highest growth rate, and the H23 showed the lowest. So on the 15th day of treatment, the H23 tumor volumes that were treated with the cisplatin polymerized hemoglobin combination were smaller than the vehicle control. The small volume was preserved on the 22nd and 29th day of treatment, and on the 22nd and 29th day, the tumors treated with cisplatin in combination with polymerized hemoglobin were significantly smaller than the tumors that were treated with only cisplatin. We can also see that on the 22nd and 29th day of treatment, there was a smaller tumor volume that was shown in the tumors that were treated with cisplatin than the vehicle group, which was 0.9 saline. We can also see a significantly smaller tumor growth in the H460 tumors that were treated with cisplatin and cisplatin in combination with polymerized hemoglobin on the 8th and the 10th days in comparison to the vehicle control, or 0.9 saline. Also, on the 12th and the 14th day, the tumor that was treated with cisplatin in combination with polymerized hemoglobin was significantly smaller than the other two treatment groups, with the cisplatin treated group coming in second. However, the A549 tumor showed very little difference until the 12th day of treatment, when the cisplatin in combination with polymerized hemoglobin treatment showed a significantly smaller tumor volume than the other two groups. So here, the cisplatin and the vehicle groups will look similar in volume. This changes on the 16th treatment day, when the tumor treated with cisplatin was significantly smaller than the vehicle, and cisplatin in combination with polymerized hemoglobin was significantly smaller than the cisplatin treatment. In this figure, we can see that the average intensity of the hypoxia-inducible factor 1 alpha and the hypoxia-inducible factor 2 alpha bands for the experimental groups. So there were no real significant differences, and there's definitely a decrease in the amount of hypoxia-inducible factors expressed, which was due to the presence of the polymerized hemoglobin. So it shows that it's working. So the materials and methods that were used in this study started with the polymerized hemoglobin preparation and analysis. The polymerized hemoglobin was prepared from human hemoglobin that was extracted from the red blood cells of, obviously, humans. The red blood cells were purified and they were combined with the polymerized hemoglobin, and all of the quantitative polymerized hemoglobin measurements were performed from the absorbance of 413 nanometers. The flow rate of the mobile phase buffer was maintained at 0.35 milliliters per minute for all of the runs with ultraviolet visible spectra, which were collected for the entire duration of each of the runs. So for cell culture and viability, the non-small cell lung cancer cell lines A549, H23, and H460 came from the American type culture collection. They were grown in single layers and they were maintained in the growth phase at 5%. So for every experiment, a minimum of 10,000 cells were analyzed. The hypoxic conditions were created using a Pro-Ox oxygen controller, which was attached to the incubator. It pulled the oxygen out. The in vitro cells were seated in 96 well plates that were incubated overnight in the hypoxic conditions, which were created using the Pro-Ox oxygen controller. And this was before they added the polymerized hemoglobin. So they were then cured for 48 hours and without additional polymerized hemoglobin and with different concentrations of cisplatin. So for the Western blot analysis portion of the experiment, the cells were treated with either a 0.9 saline, which we refer to as a vehicle, or polymerized hemoglobin, and they were incubated for 24 hours. So the conditions were hypoxic for the entirety of the 24-hour period. The cell extracts were incubated at 4 degrees Celsius for 30 minutes in a lysis buffer that was combined with protease inhibitors. The proteins were then transferred to the polyvinylidene difluoride and nitrocellulose membranes to quantify the relative density of the Western blot bands, and ImageJ software was used. And for the animal preparation, before the mice were inoculated, all of the cells were tested for mycoplasma. The female BIOL-B nude mice were injected with 5 million of the A549 cells, and the female aphthmic nude mice were injected with 10,000 of the H23 cells, or 10 million of the H23 cells, or 5 million of the H460 cells, and all of the injections were into the right hind flank of the mice. And treatment for the mice began after the tumors were 200 millimeters cubed in measurement, and the polymerized hemoglobin was given to the mice once a week, cisplatin was given twice a week. In the reactive oxygen species, diglofluorescent acetate was used to detect the formation of the reactive oxygen species. The cells were plated in a confocal dish and incubated for 24 hours before being treated with the vehicle, which was 0.9% saline. and polymerized human hemoglobin. So before imaging, the cells were incubated with the 10 micrometers of dichlorofluoroacetate. They were observed using the confocal laser scanning microscopy, and the analysis of intracellular ROS was conducted after centrifugation, and it was measured with a SpectraMax M5 spectrofluorimeter. And the annexin V propydium iodide staining, the cells were placed into 24 well-plated wells, which were plated at 50,000 cells per well, and they were left to detach overnight. So the polymerized hemoglobin with and without cisplatin were added to the culture. The cells were then harvested and centrifuged and put into 50 microliters of annexin V in a binding buffer after 48 hours. They were incubated for 10 minutes at 4 degrees Celsius and 150 microliters of annexin V binding buffer, and 20 microliters of propydium iodine were added into it. For each of the experiments, a minimum of 10,000 cells were analyzed. The percentage of the cells labeled with the annexin V or the propydium iodide were then calculated. And for the statistical analysis, the values that were graphed are presented as means and standard deviations. So t-test or one-way ANOVA was performed, which is an abbreviation for A and OVA, to determine the significance of the data in comparison to different treatment groups. So p as less than 0.05 was considered to represent a difference after all of the p-values were calculated. And so, moving on. For the conclusion, it can be concluded from the data that was calculated when the HVOCs are combined with an anti-cancer drug, such as cisplatin, the sensitivity of the tumor to the chemotherapy can be increased. So by infusing the polymerized hemoglobin, less chemotherapy treatments may be administered to achieve the same results as chemotherapy without the polymerized hemoglobin. So this can lead to reduced chemotherapeutic toxicity and improve the standard of care for patients which are suffering from non-small cell lung cancer. And the contributors to this research came from the National Institute of Health grants, the National Cancer Institute grants, and the Pelotonia Graduate Research Fellowship. And these are my references with the main article and the other references.